Thank you, Dr. Jander.
Hi, everybody.
I'm Ryan.
And today I'll talk to you about endoreduplication Arabidopsis. So in the normal cell cycle, we have the 4 familiar stages. But under endoreduplication conditions, a cell will skip M and G1 and just continually duplicate their DNA and result in more copies of the DNA than normal. This is endoreduplication. And we care about this because some of our most important crops rely on endoreduplication to form their seed. The endosperm, the most nutritious part of the cereal crops, is what we eat. And one of the main avenues of research into trying to improve cereal crop production is to try and understand and manipulate endoreduplication. However, it's really inconvenient to study endoreduplication in the cereal crops during development. They're just not available for imaging. It's difficult to work with their generation times. However, Arabidopsis provides a much better model for endoreduplication. The cells highlighted in red are so-called giant cells because they're giant, and they're endoreduplicated and elongated cells on the surface or in the epidermis of Arabidopsis. This provides us a much more easily accessible model for imaging and understanding endoreduplication. So in Arabidopsis, endoreduplication is controlled by a gene called ATML1, a homeodomain transcription factor, stands for Arabidopsis thaliana meristem layer 1. Whenever a cell is in the G2 phase, if the concentration of ATML1 protein reaches a high enough level, it passes a soft threshold, then there's a high probability of the cell endoreduplicating and elongating to become giant. You can also see this evidenced by the fact that the wild phenotype has a more or less even distribution of giant cells. A loss of function mutant, ATML1-3 mutant. Okay. A loss of function mutant, ATML1-3 mutant, will have far fewer giant cells. You'll see some because there's a homolog PDF2 acting upon endoreduplication, but for the most part, the giant cell phenotype is lost. And of course, in an overexpression line, every cell in the sepal will be giant. However, there's still some questions to be answered. How is the concentration of ATML1 regulated? It's already been shown in the Rotor lab that stochastic models are sufficient to describing the distribution of giant cells on the surface of the sepal. And this indicates to us, it's similar in a way to a reaction diffusion model. And this indicates that there's some kind of stochastic process that could be causing the decision to enter endoreduplication. However, it's unknown how and where exactly this decision occurs. Does it occur by transcriptional control or post-transcriptional control of ATML1? So you can imagine 2 scenarios. One, transcription is constant across all cells, but then the rate of degradation of ATML1 protein would be at a different rate per cell. This would imply post-transcriptional means, because if the transcription happens at an even rate, but the concentration of ATML1 protein fluctuates due to different degradation rates, this would imply a stochastic process controlling degradation after translation. In a different scenario, you could imagine that transcription is highly variable among cells, but that the rate of degradation stays more or less constant across cells. This would imply transcriptional control of the concentration of ATML1 protein. So once again, we care about this because ATML1 decides or helps the cell to decide when to endoreduplicate. So how do we intend to, or how did we study this? First, I'll explain by describing the Pp7 imaging technique. What you do is you have a gene and you insert the Pp7 sequence into this gene, preferably into an intron. And whenever RNA polymerase transcribes this gene, the mRNA transcript will have the Pp7 sequence converted into mRNA, and it will form stem loops. The mRNA will fold into stem loops, and these stem loops will then bind tightly to a viral coat protein called PCP. So if you express PCP bound to green fluorescent protein ubiquitously in the cell, the result is that whenever this transgene is transcribed, the mRNA will fold into these loops, bind to PCP, which will then fluoresce and allow you to image the transcript. To give an idea of what this looks like, these are heat shock inducible lines of Arabidopsis, courtesy of Simon Alamos. You'll notice a red background. This is HSP101 M scarlet. This is just to show the red fluorescent nucleus. And you'll notice PCP, GFP expressed ubiquitously in the plant. And these tiny little dots are HSP 101 M scarlet being transcribed in vivo. Here's another image pointing some of them out. These are the exact same image, just edited differently. To give another idea, this is taken under 20x lens, so a little bit harder to see, but this is the floral meristem before heat shock, and this is the floral meristem after heat shock. You'll notice transcriptional dots appearing. Sometimes you'll see 2 per nucleus, indicating both chromosomes being transcribed simultaneously. So what we did is we took plants in the ATML1-3 background, the loss of function of ATML1, and we gave them a transgene that contained a working copy of ATML1 bound to M scarlet, so that ATML1 would fluoresce red, with Pp7, the Pp7 sequence inserted into introns. Now we had 3 insertion locations. This allows us to handle the problem of perhaps the Pp7 loops will hinder translation, perhaps they will it will change the nature of the imaging. What this allows us to do is to reinsert a working copy of ATML1, which will fluoresce. Whenever it's transcribed, it will fluoresce green. And ideally, this will result in a plant with a working expression of giant cells, but we can track exactly when and where the protein is being transcribed. So currently, our progress has gone as thus. We've developed high quality lines that show strong fluorescence in both red and green fluorescence. This is a particular individual that showed vigorous fluorescence of both PCP, GFP, and M scarlet ATML1. In order to get pictures like this, we had to screen over 1,000 plants. So pictures like this are very nice and exciting. We selected this plant because it's strongly fluorescent and because we see a recovery of the giant cell phenotype. This is a plant in the ATML1-3 background, which has recovered giant cells as a result of the ATML1 transgene. And taking this particular individual and allowing it to self-fertilize, we have a T2 seedling that actually shows evidence of transcriptional dots. This image isn't quite as crisp because I had a higher averaging value on the microscope, and also the maximum Z projection that I'm showing here doesn't do it justice. But this dot is in fact much more intense than the surroundings, and it appears distinctly on the nucleus at a very tight level of Z value. This is another plant, another individual with strong fluorescent properties, also shows evidence of transcriptional dots, this time in the true leaves. So notice in these pictures that, different from the heat shock inducible lines, transcription of ATML1 does not appear to be happening in all cells or really in many cells at all. It appears to be only happening in select few. This leads us to a couple of hints. If transcription of ATML1 does not appear to be occurring uniformly, this might indicate that potentially the fluctuations in ATML1 protein might be controlled by transcriptional means. However, we simply need more data. Although transcription does not appear to happen uniformly at every cell at every time, it could just be transcriptional bursts happening under the normal, under normal conditions, and that post-transcriptional regulation is still occurring. In order to differentiate these two, we would just need more data. We need to take time-lapse images. We need to quantify the relationship between the transcription and the concentration of ATML1 protein. And those are some of the next steps for the project. So, like I said, some of the future goals are to develop more strong fluorescent lines, have more replicates, be able to show that this mechanism works not in just one specific, golden individual. It works in many different individuals. And once again, also to quantify the relationship between the spot frequency and intensity is allow us to track when transcription occurs and how the concentration of ATML1 protein differs after that. And one of the more broad things that our project is focused on is just understanding what ATML1 does. The exact mechanism by the way it works is not fully understood. So I'm going to give you a sneak peek into some of the computational work that we've been doing to try and answer this question. So my postdoc Vijaya, she and a collaborator, Eric Schwartz, they collected differential expression data. What they had was a synthetic promoter that would induce transcription of ATML1 after being exposed to estradiol. And so the These inducible plants were exposed to estradiol in different concentrations, 0.1 micromolar, 1 micromolar, and 10 micromolar. And then their gene expression compared to the whole genome was measured at 8 hours, 24 hours, and 32 hours. And this is just a Venn diagram showing the genes, the numbers of genes that showed up in all three sets. But for the purposes of this presentation, we'll just focus on the 10 micromolar sets. So what we found, also we should mention that in 2020, the Han research group, they, using the consensus string method, identified a binding motif for ATML1. This is very similar to an earlier publication that came up with a much similar binding motif. And searching for this binding motif, we actually find that they are significantly more likely to be found in genes in the 24 hours and 32 hours differentially expressed gene sets. This indicates to us that in those gene sets, there are genes that are targets of ATML1 and do contain this binding motif in significantly more numbers than the whole genome. This data was gathered by a bootstrapping method where we'd sample the genome 5,000 times and then perform t-tests on all those randomly sampled sets 5,000 times. And the results are significant. From these gene sets, we can produce a gene ontology. A gene ontology, for those of you who don't know what it is, whenever genes are entered into a database, a curator or the researchers, they may tag the gene with process descriptions or cellular location descriptions that allow you to describe what the gene does in a set library of terms. And so you can submit, and if you have your own differentially expressed genes, you can submit them to a computer, and the computer will ask a very simple question. Are there any terms that are more likely to show up in your gene set than if your genes were given randomly? As in, if your genes were submitted randomly, what is the probability that any of these nodes, any of these terms would appear? And if it's highly unlikely that these nodes would appear, they're considered significant. So all of these nodes are different biological processes that are tied to a gene in the differentially expressed gene set. And if we focus in on this cluster of nodes, we can see there's several nodes all related to fatty acid synthesis. We're looking at the 32-hour 10 micromolar estradiol gene set now, and you'll notice a similar cluster of nodes, a cluster of nodes all related to fatty acid synthesis. So what this tells us is that, number one, we may be seeing transcriptional control of ATML1, but we just need more data and more quantification. This also tells us that ATML1 might be involved in fatty acid synthesis. And future research will focus on trying to identify specific genes and trying to understand what pathways might be involved here. So I'd like to give a thank you to Vijaya and Dr. Roeder, my two main mentors in the Roeder Lab, as well as the rest of the Roeder Lab for giving me help and friendship. I'd like to thank our collaborators, Eric Schwartz and Simon Alamos, for helping with different analyses and providing heat shock inducible lines. I'd also like to thank Lynn Johnson from CSCU for helping me work out a bootstrapping method for understanding the significance of the binding motifs. And I believe now I can open up to questions. Diego? So like I briefly mentioned, ATML1 is thought to be the #1 determiner of entering endoreduplication and for cells to become giant. But there is a homolog. You can see here, some of these cells are still giant. There's a homolog, PDF2. I believe PDF2 acts on a different gene, loss of giant cells, correct? Okay, yeah. But ATML1 has a homolog, so you still will see some giant cell phenotype, but definitely not the wild type. So no, ATML1 is not the only one acting on giant cells, but is the primary one. Yeah, ATML1 is localized in the nucleus. ATML1 is a nuclear protein. That's actually not ATML1 at all. Let me go farther. There we go. ATML1 is a nuclear protein, so you'll see red fluorescence ATML1 M scarlet in the nucleus only, and PSEP GFP is expressed everywhere in the plant. The idea being that if it's expressed everywhere, then whenever ATML1 M scarlet is transcribed, PCP GFP will be recruited to the site of transcription. I believe I saw Jesse have a question too.
Yeah. So say we are able to manipulate this would that not have an effect on the health of the plant itself having to
Well, maybe. It has been shown in some plants, if you increase the productivity of fruit, you kind of have to sacrifice energy efficiency elsewhere. This research, though, is mostly focused on just trying to understand how endoreduplication works. And it's kind of it's a bit preemptive to say that endoreduplication in plants can't be manipulated because it will result in energy inefficiencies before we even understand how endoreduplication happens or how it's decided. Yes? No, this is all stuff we're looking into in the future of this project. As far as I know, the actual pathways of ATML1 are pretty much unknown.